Hello everyone, welcome back to another BGCSE past paper review. Today we're reviewing a biology paper, which is a paper 3 from the year 2017. And I just wanted to note that for this paper, I'm only reviewing from question number 1 to question number 5 due to time. If I do get the time, I will do question 5 and 6. Otherwise, I'll do it a different period. However, again, remember to write your school number. Always remember to do that. Your candidate number is important, your surname, and also your initials. Those are very important information to write in before you start the examination. Again, read through all instructions carefully. Ensure that you understand all instructions. And if you have any queries, please clarify those with your examiner. All right, so we're jumping right into question number 1 at this time. And this one is a diagram below show the structure of the human ear. All right, so you also need to know to label the human ear. So I'm going to go through this real quick, right, as quickly as possible due to time. And the first part here said on the diagram, label the 3 regions of the ear, the outer, middle, and the inner. So I'm just going to quickly type in where the outer ear is, and then I kind of give you the parameters in terms of where they start, where they finish. So the outer ear will be somewhere right there. The middle ear, however, will be somewhere right here. So this will be the middle here. All right. And then we have the inner ear, which is the innermost part. All right. However, again, I want to kind of draw some segments to show you exactly where they actually pretty much stop and where they start. All right. So the outer ear actually starts from the pinna, which is the outside here, all the way into where the eardrum is. The eardrum is what separates the outer from the middle from the outer. All right, and then starting from right here, let me just put it, use arrows. All right, so from there to there is the outer ear. And then the middle ear will be this section, really. So this section right here, let's come right to the oval window right here. So we actually, where the oval window stops. So it'll be right there is the middle ear. And the inner ear will start from that point onwards all the way here, okay? So now we understand the difference between the inner, outer, and middle ear, the sections. All right, so that's that for that part. Again, I'm going through as fast as I possibly can. And it said, name the 3 structures found in the inner ear. So the inner ear, which is the most inward part of the ear. And of course, there's one obvious structure that is there, but there are 2 other structures that we really need to think about. And so, and they also state the function of each of them. So the first one is the cochlea, which is the most obvious structure, and the one that I think most person will actually remember and know. The cochlea, and the function of the cochlea is really to convert vibration. So to convert vibration. All right, or you may want to say convert sound waves, convert vibration or sound waves. All right, or sound waves into impulses. That's the point. That's a function of the cochlea. All right, or impulses into impulses. All right, so yeah, so that's the function of the cochlea right there. Now the next structure is the semicircular canal. The semicircular canal is also in the inner ear as well, right? We're gonna have the semicircular canal that is also in the inner ear, and the function of that is to maintain balance. Okay, to maintain balance. All right, so that's the function of the semicircular canal. Now, another structure that is there is not drawn on the diagram, but it's not the other structure that can be found within the inner ear is the auditory nerve. All right. All right. The auditory nerve is also attached to the cochlea, so therefore it is in the inner ear as well. All right. The auditory nerve. And the auditory nerve is really to transmit impulse from the transmit impulse from the ear to the brain. And so it's not the entire optic auditory nerve is actually there, is only part of it, right? So this is used to transmit impulses, all right, to the brain. Right, and it only asks for it asks for a structure that can be found in. I'm gonna go back to the question. Let me tell you why I put this there. That's a name 3 structures found in the inner ear. All right, and say the function of each. All right, so not it didn't say structure of the ear itself, but structure that is found there. All right, and those are the 3 major structures there as well. There are other structures that are called tubules as well that are also there, and those also will help in terms of the balancing because they contain fluid. So there's a number of different structures here, but I'm keeping it basic for BGCSE. Cochlear, semicircular canal, auditory nerve. Those are 3 basic structures found within the inner ear itself. However, part C is to explain one advantage of having ears positioned on either side of the head. All right, so instead of having only one ear, like in front of you, for in the forehead or your back of your head, why you have it on 2 sides, and it kind of make plenty sense. Is really to detect a wider spectrum of sound. Let us put that right here. All right, to detect a wider spectrum. I want to say spectrum, I mean range. All right, spectrum of sound. All right, yeah, the spectrum of sound, and so that leads to, let me just say, leads to a greater level of awareness of the surrounding, right? That is important, of the surrounding. Yeah, so you get a greater awareness of your surrounding, so it can also help you in terms of your protection in your response to things in the environment. Rather than having only one ear, if you have only one ear, you're going to get sound from one side of your body, and you won't necessarily hear things coming from the next side. To help you detect a greater range of sound around you, and that's why I use the word spectrum. And when I say spectrum, I don't mean different type. Well, it also means different type of sound, but I'm talking about range. I refer to range right here. So wider spectrum or range. Let me put in this / range. All right. All right. Next part of the question here is for part D, which is the last part of this question. It said a student spins around quickly and finds that she loses her balance. Explain with reference to what happens in her ears why she loses balance. And remember, the balance is associated with the semicircular canal, right? But the semicircular canal contains what they call fluid inside. There's some fluid inside of it. So what happens really when you lose balance, that the fluid itself is distributed unevenly. So I must write it down. So the fluid in the semicircular canal, canal, all right, becomes unevenly distributed. All right, unevenly distributed. That's simply what happens. So the liquid or the fluid within the semicircular canal becomes unevenly distributed. And so as a result, no. All right, so this will send a sudden signal to the brain, indicating, send a signal to the brain indicating that there is let's say lack of equilibrium. All right, so the fluid is no longer in equilibrium, so of course you're going to create some unbalance, and you lose, and you feel like you're losing balance because the shift in the fluid actually sent to your brain. Your brain interprets that, and you will move according to how the fluid is moving. And remember, if you think about like a jelly, if you touch a jelly substance and shake it, you notice the liquid starts shaking. So your entire body will kind of have that same type of effect. You're going sideways, back and forth, because the fluid in your brain, in the air, is actually moving. That type of motion is interpreted by the brain, and your body respond according to try to balance back the fluid. All right, so you moving back and forth is actually trying to balance the fluid and to get your stability back. All right, so that in that part of the question 1. All right, so for question 2 now, so the table shows the concentration of 2 gases in the atmosphere between 1980 and 1990. You have the gas named carbon dioxide and nitrous oxide. It's a concentration in atmosphere in part per million in 1980 and 1990. Here we have 290. We have 0.2 for the nitrous oxide. We have 354 for carbon dioxide, and we have 0.3 for the nitrous oxide. And percentage change of gas over time period calculated from the original. For nitrous oxide, we have 0.07. There's a missing section here for the carbon dioxide. So let's see if they ask us to calculate that. But more likely, though, we have to calculate that thing. All right, so part A is said what trend is shown by all the data for these gases between the time period 1980 and 1990. So 1890 and 1990, right? So 1890 and 1990. What trend do we notice here? So if you move from 1890, you see 290, and from 1990, you see 354 for carbon dioxide. For nitrous oxide, you see 0.2, and then it goes up to 0.3. That means there is an increase there. So the concentration of gases in the atmosphere increase over time. Okay, so the concentration of the gases increase. All right, increase gases increase over time. All right, so over time you get more gases in the atmosphere. Okay, so that's pretty much when you say increase, then that's actually your mark. All right, so the concentration of gases increase over time. Here, asks us to calculate the missing percentage change value in the table. Show you working. So they ask us now to calculate this missing value here. And matter of fact, I could do it in chart because it's going to be easier for you to see here. And so what you're going to do is the difference, find the difference first, and then the change is going to be over the original. So what you're going to do is the original is 290. Okay, the original value is the 290. That's where it's coming from, and the final value here is 354. So you're going to minus those values, and so you're going to say 354 minus 290. All right, which equals to 64. So you're going to 64 is the answer there. And you're going to divide 64 by the original now. So when you get 64 divided by the original, which is 290. And then you're going to multiply by 100. So all of that, you're going to multiply by 100. All right, so multiply by 100, get your percentage. All right, so if you notice, they have theirs in decimal. So I'm going to put it in decimal as well. So just do not multiply by 100 here because they have a decimal. What you should get is 0.22. Okay, same thing as 22%. The 0.07 is the same thing as 7%. They didn't actually just change it to percentage. They have it in decimal. And so you could write the percentage or decimal, not a problem. So based on the calculation here, the answer here is 0.22 or 22% different. All right, so that's the difference right there. All right, so for part C now, it's a state one reason for the difference in percentage change between the two gases. So what is one reason why there's a difference in percentage change between the two gases? All right, because one is carbon dioxide, one is nitrous oxide. And so because you have a greater amount of carbon dioxide, what I'm going to say right here is that what caused the difference here is simply because you could have more combustion of fuel. So you can have a more combustion of fuel. Because when you combust fuel, you get a large or a great deal amount of carbon dioxide. So you have more combustion of fossil fuel over the years. So that could be a reason right there. Another possibility here, it could also be greater value of or greater level of deforestation as well. So you have greater levels of deforestation. And why I put in this here, because deforestation, if you think about it, if you have less trees, then definitely going to have less carbon dioxide, because that is the major ways in which carbon dioxide is removed from the atmosphere, right? So greater levels of deforestation. And when you have deforestation, it's simple. You can have more construction, you can have more development and all those things. So that's also the reason as well. To take down trees is to build houses, to build buildings, just to kind of develop physical infrastructures. All right, so while we build up nice buildings and pretty places, we also have a strain on the environment as well, because we have a buildup of carbon dioxide because we have less trees. All right, for part D now, it said explain how an increase in the concentration of carbon dioxide in the atmosphere can lead to global warming. And this first thing to mention here for 2 points, I want to talk about increasing carbon dioxide. How can it lead to global warming? And first point I want to make is that that carbon dioxide is a greenhouse gas. Okay, so the first thing I want to make is that carbon is a greenhouse gas, which means it's going to be in the right at the ozone layer, all right, and is capable of trapping heat. All right, so it's a greenhouse gas. And so next point I'm going to make right here is that light rays, can pass through the gas easily. All right, so light rays can pass through the gas easily. That's first thing. Can pass the gas easily. When the light hits the ground, it converts to heat. Okay, so when light hit the ground, it converts into heat. All right, and so when light is converted to heat, then what happens next is that the radiated heat. So when they convert to heat, the radiated heat, now because they're going to radiate from the earth, the radiated heat will reflect. Because radiation is kind of reflection, reflects the radiated heat from the earth. Let me say the radiated heat from the earth, all right, will get trapped, trapped in the gas, which is carbon dioxide. The gas, which is carbon dioxide, that's what we're talking about right here. We're trapping the gas, which is carbon dioxide, because light could pass through, but heat cannot pass through so easy. All right, and that's the reason why it traps the heat. Get trapped in the carbon dioxide. All right, we get trapped in the carbon dioxide. All right, so because of this now, so this result in an increase in temperature, all right, or heat. This increase in temperature of the Earth's surface, okay, of the Earth's surface, all right, or atmosphere, all right, surface / atmosphere. All right, so simple that what is happening there. Light could pass through the carbon dioxide really easily, and then but when the light hits the ground, the light converts into heat energy. The heat energy is radiated back into the carbon dioxide or pockets of carbon dioxide or blanket of carbon dioxide, and the heat is not able to pass through it to go back up into space. So instead, it gets trapped and remains above the Earth's surface, which is in the atmosphere, and the atmosphere gets warmer. And of course, because the atmosphere gets warmer, then the entire Earth itself will become much warmer, and that is a phenomenon called global warming. All right, next part of the question here, and this is last part of this question. It says, state 3 consequences of global warming. There are 1,100,000 consequences of global warming. There are too many to be named. All right, so global warming. So think about it now. Our actions is what causes global warming. Our actions. So flooding. Flooding is one, right? And flooding will cause rising sea level. So rising sea level. All right, so I'm just telling you this. I want you to be very afraid. I want you to be really afraid of global warming, because we should be afraid of global warming. Maybe we don't know. We also will have frequent or extended droughts. Okay, so we have frequent or extended droughts. All right, we're definitely going to have that. Also, what we're going to have is, again, we're going to have warmer ocean. So we're going to have warmer ocean. All right, and if you have warmer ocean, then you're going to have chloral bleaching, and a number of different things going to start happening there. So we're going to have chloral bleaching as well. And once chloral bleaching starts taking place, it's going to affect the other organisms. You're not going to have any lobster, any crawfish, any snapper, any parrotfish, any grouper. All those will be affected. So I want you to be very afraid. In fact, you may not have food. persons may even lose their jobs. So global warming could lead to so many things. It could cause even houses to wash away because of high levels of sea level, and also because of excessive flooding and all those type of things. So be very afraid. Be very afraid. Our actions of being selfish and dangerous to the environment could cost us our life. All right. What we can also have again, we could have more wildfires. See, more wildfire or forest fire, or wild or let's say forest fires. Right. We can have more of that as well. All right. So yeah, be very afraid. Yeah. Still, I'm gonna give you some more. I'm not finished yet. I want you to be really, really afraid of this because it's a very, very serious thing that most of us don't take serious. Or simple actions cause these dangerous, detrimental effects over a period of time. So we have more fire. We also have migration. So migration of animals as well, or organisms. All right. I say organisms. Yeah, some of the organisms that we know, we're not going to see them again. That's it. They're gone. They either gone somewhere else to live, or they become extinct. We can also put that as, again, let's put that extinction. Yes, extinction of organisms. So when it get really hot, like your artificial selected breeding dogs, like you Shih Tzu or whatever dog you have, they cannot bear the heat and they may not die. You're burning more electricity because of you have to use more current for AC and all that, and then they contribute even worse to the global warming. All right, so definitely be very afraid. Be very, very afraid. I'm not finished with this because a matter of fact, global warming is like hell. Yeah, it's like we're creating our own little hell on Earth. Yep. Destruction of habitat. Destruction of habitat. Yeah. So yeah, it's very serious thing, and even destruction of habitat, destruction of your homes too. You may not have nowhere to live because your buildings start to damage, and when rain comes, then water start coming in and all type of thing. Another thing here, farming. You may have also farming as well, that we cannot find food, and since you cannot find food, then we may have what, starvation. So we may have starvation as well, lead up to starvation. And if people cannot find food, then what happen going to happen? Have social problems as well, and people will start fight for food. People start hunt other people to get food from them as also. Yeah, so be very afraid of global warming. Be very afraid. Our own actions is what causing the problem. So we need to be very afraid. Yeah, we need to have fear in global warming. We don't take it serious, but it's a very serious thing. It can cause us so many things. The things that you know now may be lost because of global warming. All right, question number 3. Is a sickle cell anemia is a common genetic condition found in many Afro-Caribbean populations. What is the genetic cause of the sickle cell anemia condition? So the genetic cause of this is simply a mutation of the hemoglobin gene. All right, it's a mutation. There's a mutation of the hemoglobin. All right, so hemoglobin globin gene. All right, so the genes are responsible for formation of hemoglobin. The gene is mutated, and as a result, the number of things happen. So you say, how does this condition affect the blood of an individual? All right, so let's give you the effects right here now. So what it does is cause hemoglobin molecules to join together. So let me just make a point right there. So hemoglobin. All right, so hemoglobin molecules, let's say molecules. And hemoglobin is a type of protein, just to make a mention of that. So hemoglobin molecules are proteins, stick together. So they stick together, all right, stick together, resulting in abnormal shaped red blood cell. Okay, abnormal all right, abnormal. Okay, I'm missing something. Abnormal shaped red blood cells. All right, red blood cells. Yep. So the red bloods become abnormally shaped, and not only that, and because they become abnormally shaped, they are unable to transport oxygen effectively. All right, so I can say this result in a decrease in efficiency. This result in a decrease in the efficiency, all right, in the efficiency to transport oxygen. All right. All right, so that's what happened to the red blood cells. They become abnormally shaped because the hemoglobin stick together, and of course, because of that as well, they were not able to carry out transport oxygen effectively as well. And because of the shape of the red blood cell, they kind of have a crescent shape, right? And so sometimes they get stuck in the capillaries as well, and they lead to pain and all other different types of effects as well. All right, the next part of the question now said the family tree below shows the inheritance of condition sickle cell anemia over 3 successive generations. And I just want to say that this is also called a pedigree chart. So I'm going to show you the pedigree chart. All right, that's what it's also called as well. So if you notice, the circular ones represent, the circular shapes represent female, just to point that out. The square ones represent the males. So males are square, females are circular. All right, so the code there now is that unshaded sections, they are normal, male or female. If it's fully shaded, that means they're affected, that means they have the sickle cell fully. And if they're off shaded, mean that they are carriers. And there's a point out that carriers mean that you're heterozygous, right? You're hybrid. All right. And since sickle cell anemia is a recessive, matter of fact, sickle cell anemia is recessive. So therefore, if a person to have it fully, they must have 2 recessive alleles, just to kind of mention that. So the first generation right here, the first generation, just to point out, this line actually means that they are married, right? This line means that they are married. So horizontal lines across the shapes mean that they are married. All right, and so you notice male, female in all of these cross-section combination. And then the horizontal ones now, they mean offspring. So the married couple that having children are the horizontal ones, so that means marriage. And we use the word marriage. You're not saying that you only can be married to have children, but that's just a typical situation. Well, that's how it should have been. Everybody should get married before they have children. That is the ordained principle by God. However, it means producing children. It means mating generally by marriage. All right. So if you see it in the exam, it really means marriage or mating. And the vertical ones mean offspring. So coming down like this down the line, it means that they're offspring. So like here is an offspring of the first generation there, offspring of the first generation, offspring of the first generation, offspring of the first generation. And then this first generation, now which is second generation offspring, they will now marry to other persons. So this is a person you've been married to, the first generation female, which is a carrier. This person here is marrying to a normal person, a normal male, marrying to a normal female from the first generation, which is, and the children of the first generation becomes the second generation. All right, so you get the drift with marriage and offspring. All right. Now, that's the basic bottom line you need to figure out there. Marriage, offspring. So let me just take these back out. I may need the space to do something. I think I need all these spaces. Let me take them out just in case. I just want to understand the difference between marriage and offspring. Vertical is offspring. Horizontal is marriage. Vertical is offspring. All right. So here now, say use the letter. This first part question. Part B. So where's part A? Hold on. I think I missed something then because this is part B. Oh, okay. So part A we did. Okay, so number 3, part A. So part B now. Okay, I thought I missed something in this question. All right. So for part B now, we say using the symbol uppercase H and lowercase h, identify the genotype for a sickle cell anemia condition. So we're looking for the genotype. A genotype is a combination of alleles. And remember now, if uppercase and lowercase identify the genotype, so if you have sickle cell anemia condition, it therefore means you have to have 2 small h's because sickle cell anemia is a recessive disease, all right, caused by a recessive allele, I should say. And now if you have sickle cell trait, which means you're a carrier, which means you have the trait, you have the allele, but you cannot show it, which means you're going to mask by a normal, so the uppercase H is a normal, so you're going to be that. And for a normal red blood cell, which means no form of sickle cell at all, you're going to be 2 uppercase H's. All right, so that's the genotype there for the 3 different conditions. All right, so let's go down now into the next part. It's a using a genetic diagram, right, to illustrate, determine what the possibility is of male W having a sickle cell trait with if he marries a sickle cell trait woman. All right, so let's make sure we understand this now, right? It said that the male W, let's highlight that, the male W. It said the male W, let's find male W in our chart. All right, let's go to male W. All right, there's okay, there's no male W at all, right? There's no male W. All right, no, all right, there's no male W. So I don't know which male you're talking about. All right, so this may be a mistake in the exam, but what I'm going to tell you, right? Let me just look through this and show what some things that you need to know. All right, so this person is a carrier. I'm just going to show you this. There's no male W at all. And all right, so that this person is that, we know that for sure. This person is going to be 2 uppercase H's, that's normal. All right, what I want to look at is the males in the entire family tree. You have a normal male here, a normal male here, a normal male here, a normal male, a normal male, a normal male. So you have a lot of normal males within the family tree. And then we have some carrier males. So we have no males with the sickle cell. So males are either carrier, based on what I'm seeing in the chart. Males are either carrier or they are normal. But I want to go back to the question again because I want to make sure I'm not missing anything because I've not seen a male W at all. It's a using genetic information, using genetic diagram to illustrate, determining what is the possibility if male W, which we have male W for real, having a sickle cell trait, marries a female with a sickle cell trait. So we're looking for the child. So the child now having sickle cell trait. So notice we said now to determine what possibility is of a male having a sickle cell trait child. So I want to know what's the possibility of having a sickle cell trait child. All right, so that's what we're looking for, a sickle cell trait child. But as I'm saying to you, there's no male W. But males can only be of 2 parts, right? 2 different types of male. All right, so let's tell you what males we can have. We can have male that is either, so we have 2 options. So let's do this. The only thing we're sure about is the female. Okay, we're sure about the female. The female is sickle cell trait. And if female is sickle cell trait, then that she is an uppercase and lowercase h. That's what we're sure about. That's the only thing we're sure about, as a matter of fact. She is that. The male is of 2 examples, of 2 types, one of 2 types. And again, I'm going to do the 2 of them because, again, there's no male W in the chart. So on one hand, I'm going to do the male. I'm going to use the male being this. So the male, let's put the male over here. So the male is going to be, in this case, uppercase H, because all the males in the chart, they are either normal or they are carrier. And the female now, the female is one thing. The female is heterozygous. Based on the question, it said that she is a carrier. All right, so she's going to be that. And then that other option now, so I'm giving you 2 different options. The other possibility for the male is that the male could be a carrier based on the chart. So we'll have 2 possibilities of males. And the female, again, she is heterozygous. That's the only thing we're sure about. So we're going to draw our Punnett square now and determine what is the possibility of having a child that is sickle cell trait. Okay. All right. So let's get down into drawing a Punnett square. All right. And by now you should be kind of good with this. You're kind of good with this because you can guide yourself from here to draw your Punnett square. So your Punnett square will look something like this. The Punnett square. Let's take it down the line. All right, let's draw another Punnett square over this side. I'm just going to do them side by side. All right, that's a Punnett square. All right, fast, nice, and easy. All right, so let's put on now our characteristics things. Let's put this in blue, motherfucker. All right, so let's put on that. Let's put the male on top here. So you have male is uppercase H, and the female here now is uppercase H and lowercase h as a female. All right, let's put this on the same box over here as again. The female is the same, but we know the female for sure. But the male now, the option for the male is also heterozygous, because remember we only have 2 options of male in the chart. Here is 2 uppercase H's. Here, 2 uppercase H's. Uh-oh. Let's see if I could get that out. All right. Okay. All right. 2 uppercase H's. All right, so two uppercase H's there. Here is also two uppercase H's as well. I know some of you don't need to go through this long step because as you look at it as a genotype, you know exactly what you get as a result, right? Here is a uppercase and lowercase. All right, so it's good if you could master this: uppercase, uppercase, and lowercase as well. Here we have uppercase. On this side, we have uppercase. And we know that when we have two heterozygous parents, we have a 3-to-1 ratio. So it's something that you should know. Two heterozygous parents lead to 3-to-1 ratio. So this is uppercase, lowercase as well. All right. And here we have two lowercases here. What do you know by the result right here is that from the first possibility, the first option, if the male they were talking about was normal, then these two that form at the bottom right here, let me just put this in a different color to circle this, so we're going to know exactly what we're talking about now. These two males are possibility of carrier, and we say looking for offspring that are carriers. So that's carrier right there, and this option here, these two are carriers as well. So we notice in both situations, based on the males that exist within that pedigree or family tree, no matter what the male do, once any of the male have children with a carrier female, the possibility of the offspring is the same, right? 50% chance. So there's 50% chance being a carrier or a child heterozygous trait. So here is 50%. That's 50% chance. 50% chance of having a child that is a sickle cell trait, or otherwise called a carrier. 50% chance. All right, so let's get into the next part of the question now. Is the last part of this question. State the term used to describe two alleles of a gene where both affect the phenotype of a heterozygous individual. So if both alleles affect the person in a heterozygous situation, we call this thing codominant. Okay, so this is called a codominant type of thing. All right, a codominant, that's the term used to describe if both alleles that are different affect the phenotype. All right, so for example, you may see red and white. All right, showing up. And a blood group, for example, the AB blood group, yeah, A and B together give a AB. All right, so this question, jump to this part of the question. Question number 4 now. And question number 4 says the diagram below shows an experiment to investigate how light intensity affects the rate of photosynthesis in pondweed. And this pondweed is called the Elodea. All right, so Elodea is the name of this pondweed. All right, so let's just go into this and point out a few things first. All right, so we have the bubbles here, and bubbles giving off, we know what bubbles that should be is oxygen because the plant is photosynthesizing. However, you have a lamp right here that's producing a certain amount of light, and we have a 10 centimeters incremental across the ruler. So now this lamp move away by 10 centimeters each time, 10, 20, 30, 40, and so on. All right, so it's a regular Dex lamp. All right, and then what we have here is a rather balanced chemical equation for photosynthesis. All right, so a balanced chemical equation for photosynthesis. Everybody should be expert in doing that by now. Plants require carbon dioxide, so you're going to have CO2 for sure. And CO2 going to react with H2O. Or matter of fact, I'm going to say 6 CO2 because everything will have 6 in front of it except for glucose. It reacts with 6 oxygen. Let's call that 6. I don't like that. Take this out. All right. I was writing oxygen first. But there's 6 oxygen, so going to be 6 oxygen, 6 O2. And it will give you your, let me put this arrow long, it will give you your carbon, your glucose, which is C6H12O6. So C6H12O6. All right. And also what you get is oxygen, and oxygen will be 6 molecules of oxygen, going to be plus 6O2. All right. And so that's the basic idea right there. What you're going to have here is also light is needed as well. So you can have light on top of the arrow, and on the bottom of the arrow, you're going to have chlorophyll. But light is definitely needed. Chlorophyll should be there to absorb the light as well. So chlorophyll on the bottom of the arrow, and that will be your balanced chemical equation there, indicating photosynthesis. All right, that's something you need to practice, something you need to know. All right, there's a state why the water temperature in which the weed has been placed should be kept constant during the investigation. And it's something of a control. It's called a control variable, right? But why? The question that asks what it is, they say, why you keep it constant? And remember, you're only testing, you're only investigating distance of light from the weed, right? If we go back to the question or the statement, the experiment is all about investigating how light intensity is affected by the rate of photosynthesis, right? So light intensity, which means and the further away from the light you are, is less light coming onto you. So you're testing the light intensity, not let's go back to the question. Let's say temperature, right? So the temperature of water is not what we're not testing what temperature affect photosynthesis. We're testing light. So why we keep this constant is to ensure, right? Is to ensure no variation in temperature, all right, or to ensure that the only thing that is affecting the rate of photosynthesis is light. All right, so to ensure that only light is affecting the rate of photosynthesis. Affecting the rate. Let's put the rate of photosynthesis. Okay. Yeah. So we want to make sure that only light is affecting that rate. All right. Only the light. So by moving the light, that is the only thing that is changed. So you want to keep everything constant, and the only independent variable that should be there is the distance of the light. And what is going to respond to that is the rate of photosynthesis, which is going to be the dependent variable. All right, if we go to part C now, so the table below shows some data collected from the investigation carried out. And so we have the investigation, the data right there. We have the distance of lamp in centimeters, the number of bubbles produced in a minute, and then we have a graph that's the data right there. But I skipped through that and I quickly draw the graph for you to make life easier on us. So this is a graph right now being presented here. I want to point out a few things. Notice there is a labeling on the axis. You have to label the axis as well. And the axis is giving us the number of bubbles. And then on the x-axis, we have the lamp distance in centimeters. And we use appropriate scale to make sure that our graph is nice and big onto the page. Also, what is important too, you need to use fine points to make your points or dots or coordinates, and then use straight edge, which is a ruler, to make a line fine and smooth. That's what they're going to get the marks for. All right, the line should not be zigzag and all that. Here it asks us to predict the distance of lamp at which the number of bubbles produced would be 14. All right, so from this now we're going to look at our graph carefully, and then we're going to match up with lamp distance. I want to find the lamp distance. It said predict the distance of the lamp at which the number of bubbles produced would be 14. So I want to match up where 14 is. So if we come on the graph here, 14 will come across here like that, and then all the way down to this point. So that's going to be 50. So 50 will be our value when the number of bubbles is 14. So here is 50 based on the graph. All right. All right. So next part of the question now, we said describe the effect on the number of bubbles of moving the lamp further away from the pondweed. And so the further away you go from the pondweed, what you realize that the number of bubbles decrease, all right? Because decreasing light intensity, decrease in photosynthesis. That's the reason why. That's the reason behind it, okay? But here the number of bubbles decrease, okay? So decrease in number of bubbles. All right, so decrease in the number of bubbles. Less bubbles are formed, all right? Decrease number of bubbles. All right, the next thing is the state of the change in bubbles production between 20 centimeter and 30 centimeter light distance differ from 40 and 60. So look at 20 and 30 and 40 and 60. What's the big difference between 20 and 30, 40 and 60? All right, I can even go to the chart and show you. I could do it, look at the graph. I'm going to do it 2 ways. All right, so 20, 30. If you look at 20 and 30, from 20 and 30, you move from 25 to 24. That means you decrease by 1 centimeter, 1 bubble, right? However, from 40 to 60, it moves down by 8. So you lost 8 bubbles. All right, so if you notice the difference there, it only decreased by 1 between 20 and 30, but between 40 and 60, it's by 8. So it's a greater decrease in the number of bubbles. So let's see how we're going to answer this now. All right, so let's go back to this part of the question here. right here. And it's the last part. All right, so it says show how the change, it's a state how the change in bubbles production between 20 and 30 centimeter light distance of, differed from that between 40 and 60. All right, so what the first thing we can talk about is that between 40 and 60, so 40 to 60 experience a greater incremental change. All right, so experience a greater incremental change. All right, so which means that the changes between them is greater. All right, greater incremental change. All right, compared, let's say compared to 20 to 30, okay? To 20 to 30. All right, so the bigger change, the greater change. All right, that's one thing. Now, the thing is, let's also give a reason now. And it's a state, that's one mark. So the incremental change is bigger, but let's talk about why is this so. Between 20 and 30 is a slight change. That means the light is still relatively close to the plant, right? So since the light is relatively close to the plant, then it's going to photosynthesize almost the same rate. When they go further and further away from the plant now, the light intensity towards the plant is getting really, really less. And so even a slight change in distance will give a bigger change in number of bubbles as well, right? Because it is almost far apart, so far that it has almost little to no effect at all on the plant. All right, so this is the end of this part of the question here. And now we're going into section B. And again, I'm only going to answer number 5 in interest of time because I have some other stuff to do as well. And then hopefully I could do 6 and 7 on this paper. If not, I'll do it another time. But definitely look out for that. All right, so let's go in the second part of the questions. So section B here, question number 5. And this question actually reads that the phylum Arthropoda is one of the largest on this planet. I don't know if a different planet may have more arthropods, I don't know. But it's on this planet you have the greatest amount. Maybe we need to check Mars or one of them places to see if we have more arthropod. All right, so it contains 4 separate class, I should say, classes. So there are 4 separate classes with many commercial important species in them. All right, so we're on planet Earth, and we have a lot of arthropod on this Earth, on this planet. So maybe a different planet maybe have a little bit less or a little bit more. I'm not sure. I'm joking though, you know that, right? All right, part A is a state 3 features common to all arthropods. Or the 3 features common to all arthropods is 1, that they have exoskeleton, they have jointed legs, they have segmented body, and they have no backbone, which means they're all invertebrate. They're all invertebrates. All right, so part B now. Part B is to name 3 of the classes in arthropod phylum, and for each class you named, list 2 external characteristics specific to that class, and that's a 9-mark thing. And so insect is 1. An insect has 6 legs, 3 body segments, and the body segments are head, thorax, and abdomen. That's very important. They have 1 pair of antennae, and they have either 1 or 2 pairs of wings. Okay, so insect will have 1 or 2 pairs of wings, but generally though, insects have 2 pairs of wings. Just to point that one out. All right, crustaceans, and we love our crustaceans. We all know crustaceans, right? All right, so crustaceans now, which include, of course, the lobster and so on. All right, so crustaceans will be 10 to 14 legs for the most part, 10 to 14 legs. They have 2 body segments, and they only have 2 pairs of antennae, okay? All right, for arachnids now, arachnids like spiders, if you're Spider-Man, yeah. So you have 8 legs generally. Well, they have 8 legs typically. 2 body segments, no antennae at all, and they have no wings. All right, and I'm giving the 4th one. They will ask us for 3, I think. All right, so we have myriapods, and myriapods either of 2. We have diplopoda and chilopoda, and the diplopoda and chilopoda will be millipede and centipede, right? And in a combination, they have between 28 to 200 legs. All right, from 28 to 200 legs. Of course, millipede and centipede will be different. And then they have up to 14 to about 50 body segments. All right. And so in one case, you have only one pair of legs on one body segment. Another situation, you have two pairs of legs on one body and one segment. So it depends on which one you're talking about, whether Diplopoda or Chilopoda. But the fact that you have a lot of legs, you have a large amount of legs. If you don't remember the numbers of legs, that's fine. So what you can just say for myriapods or myriapods is that myriapods, they have a larger number of legs and also a large number of body parts, and you're good with that. They will accept that. All right. Part C now is many of the commercial important arthropods, such as the spiny lobster, and this is the Panolirus argus. It said it inhabit the coral reef of the Bahamas. Is a state two reasons why the coral reef is a suitable habitat for the lobster. And so one of them is that the crevices, the crevices they could hide. They're supposed to say they have crevices to hide from predators. So they can have the crevices within the coral reef, like small spaces, and looking at the rock structures, they could hide in those spaces. The calcified part of the coral reef, they could hide up in those little corners and little space, and predators won't get them. Also, on the coral reef, a lot of food sources as well. So you find them on the coral reef, they stay there for shelter, they stay there for also food as well. And so they eat organisms such as sea urchin, all right? And so sea urchin will be a lot of sea urchin on the coral themselves, because urchin love to be on those rocky structures, right? Or rock-like structures. And also the spiny lobster, if you notice the color of them, they can also camouflage on the coral reef as well. So that's three good reasons there. They have protection because they can hide from predators within the crevices of the rocks or the rock-like structure. They have a lot of different food sources there, and also they can easily camouflage with the rock-like structure or rock-like part of the coral reef. All right, for part two now, it's a state one problem caused by nature, which the lobster faces living on the coral reef. And so one, you have dilution of water, and this is because of excessive rain. So you have excessive rain, it can decrease the salinity of the water. And of course, you know that organisms like a certain level of salinity, otherwise they cannot reproduce, they cannot even absorb water, and their cells could also be damaged. All right, and so some organisms, they are very sensitive to the salinity or the salt level in water. Another big thing here, which is naturally occurring, is competition. So competition is there because of large and diverse ecosystems. So they compete for food and all of those things, right? They even compete in terms of living space because other organisms want those crevices to live in as well. So yes, another big one here is predators. All right, so predators, they're natural predators. The organisms feed on them. And again, because the ecosystem is so large and diverse, then you realize that the feeding relationship becomes more intensified as well, right? All right, natural disasters such as hurricanes, and hurricanes would cause heavy wind, not heavy winds. It's supposed to every waves have every wind, but it's supposed to every waves. So every waves come from hurricanes. All right, land will experience the wind, under the ocean will experience the waves. All right, so every waves from hurricanes. All right, so that could cause a big problem for them. They get battered by the waves. Not only that, the waves break down part of the coral that they actually use for shelter, and so there are a number of different things happen. I mean, hurricane could also damage your food sources, damage habitat, and all of that. All right, so loss of food due to coral bleaching as well, because that could also be a phenomenon caused by different factors such as climate change, diseases as well could also be a big problem for them as well. All right, so again, part three is the ocean acidification as a result of global warming is one of the major man-made problems affecting coral reefs. Describe fully what we mean by ocean acidification, its specific causes, and its key consequences on the corals. So on the corals, that's a key thing here now, on the corals. Not just on the ocean, but on the corals. Not on any other organism, but on the corals. So we need to be very specific when you ask these things. So be careful, read carefully, make sure you understand what they're actually asking you, right? So acidification is simple, the gradual decrease in the level of pH of the ocean. So in other words, in basic term, is that the ocean become more acidic. The ocean is forming or becoming acid. That's what it really means in a layman term. The causes of this is simply dissolve of carbon dioxide. When carbon dioxide dissolves in the water, it forms acidic substances. All right, so form what they call carbonic acid, right? And also indirect causes, because of production of carbon dioxide is simple because of burning of fossil fuel, we know produce a lot of carbon dioxide for that. Deforestation is also another cause for ocean acidification, because less trees mean that more carbon dioxide, water will have to absorb some of these carbon dioxide, and hence it can let cause the ocean to become more acidic. All right, but the general thing here is the absorption of carbon dioxide within the water body. All right, so consequences now is that it degrades calcium carbonate, because if the water is acidic, it reacts with calcium carbonate or any form of carbonate in the water, and deplete them. So it will form even more carbon dioxide, because when carbonate react with acid, you get carbon dioxide, salt, and water. So you even produce more carbon dioxide within the ocean itself. And just to make a point right here is that that things like the conch, lobsters, and so on, they absorb calcium carbonate, to make their shells. But again, coral also absorb calcium carbonate with limestone, and to make their exoskeleton as well. So we're specifying right here to the coral effects on the coral. All right, so coral definitely need carbonate to make their exoskeleton. Again, within the coral polyps, we have zooxanthellae, and of course, if water becomes acidic, it will damage the zooxanthellae, and the coral will bleach, and them themselves will die, because there's a very important symbiotic relationship between zooxanthellae, which is algae that live inside the coral polyp. The zooxanthellae provide a nice color, provides oxygen for the coral as well, because the coral is animal. Just to mention that the coral polyp is animal, right? And the zooxanthellae is algae, which is a plant-like. So just like an animal and plant living together, they benefit from each other. All right. All right. So, all right. This is where I stop for now. Again, as I say, I'm stopping at question number 5 here because in interest of time. Hopefully, I get the chance to do the others. Again, thank you for being here. Thank you for being here. Good luck on the examination. See you soon. All right.